Morning, my dear students. Welcome to our Zoology online class. Hope you are very happy. And dear students, in the last class, we studied about two important topics. The first topic is erythroplastosis fetalis. Okay, it's a disease. So we studied about this erythroplastosis fetalis disease and how we can prevent this disease. And the second topic is sex determination. How the sex is determined in some organisms. Okay, so these two topics we have completed in the last class, dear students. So today's class. Okay, so today we are going to study about some more topic. So listen very carefully. So first we are going to study about sex-linked inheritance. Okay, so today our first topic is sex-linked inheritance. Sex-linked inheritance. So in the last class I told you in human body all the cells consist of 46 chromosomes. First 23 chromosomes, all these 46 chromosomes are arranged in 23 pairs, and the first 22 pairs of chromosomes are determining our body character, and the last pair of chromosomes are called sex chromosome. That two chromosomes are determining our sex. Okay, and each and every chromosomes consist of a particular number of genes. Listen very carefully. So the first chromosome consists of a particular number of genes. Last chromosome consists of a particular number of genes. Like that, each and every chromosomes consist of a particular number of genes. Okay. Here the sex chromosomes. Sex chromosomes you all know. In female, two X chromosomes are called sex chromosome, and in male, one X chromosome and one Y chromosome are called sex chromosome. Here the sex chromosomes also consist of some genes. So the genes present in the X chromosomes are called X-linked genes, and the genes present in the Y chromosome are called Y-linked genes. Okay, hope you understood this part. So genes present in the X chromosomes are called X-linked genes, and genes present in the Y chromosomes are called Y-linked genes. Understand a few? So first you are going to study about inheritance of X-linked genes. Okay, inheritance of X-linked genes. And two examples are given in your book for these X-linked genes. So the first example is hemophilia and the second example is color blindness. So listen very carefully, dear students. These two diseases are togetherly may be asked for 5-mark question and separately may be asked for 3-mark question. So listen very carefully. So now we are going to study about the first example for the inheritance of X-linked genes. Hemophilia. This hemophilia is otherwise called bleeder disease. Hemophilia is otherwise called bleeder disease. And this disease maximum affect the male than the female. Okay, so the hemophilia disease maximum affecting the men than the women. Second point. And the third point, this disease was first reported by one scientist. His name is John Cotto. He only first explained this disease in the year of 1803. Okay, so John Cotto, the person, John Cotto only first identified and explained about this hemophilia disease in the year of 1803. Okay, and the next point. Actually, our blood consists of a substance. Name of the substance is called thromboplastin. So, if any injuries occur in our body, immediately within few minutes, the blood is clotting in that area. This is because of the presence of one substance. That substance name is called thromboplastin. Okay, that substance name is called thromboplastin. And some genes are responsible for producing this thromboplastin substance. So if anyone is affected with this hemophilia disease, the thromboplastin substance is unable to produce in their body. Okay, so if any injuries occur in their body, there will not be any clot. The blood will not clot. So continuously blood is bleeding. So once I am going to repeat this point. See, thromboplastin is the substance present in our blood, and this substance only responsible for the blood clotting, and this substance, the production of this substance is controlled by some genes. So if any problems occur in these genes, or if this gene is involved in mutation, the gene is unable to produce the thromboplastin substance. So, if any minor or small injury is happening in their body, in the affected person's body, the blood will not clot continuously bleeding. Understand if you understand? And the females only always the carrier of this disease. Carrier meaning transferring from one generation to next generation. And the female, 50% of the female transmit this disease to their son. Okay, 50% of this disease is transmitted to the son from the carrier mother or carrier female. Understand a few? So dear students, that's all. Then the last point. This disease is one of the example for crisscross pattern. Crisscross pattern. So that's all about this first disease came out here. So once I repeat all the points. So the first disease by this it's a bleeder disease. It is otherwise called bleeder disease. And this disease is maximum affecting the men than the women. Second point I said. And the third point is this disease was first reported by John Pato in the year of 1803. And the next point, fourth point is thromboplastin. I said about the thromboplastin. Here thromboplastin is used for blood clotting. So if anyone is affected by this disorder or this disease, the thromboplastin production is completely absent. So the blood is not clotting, continuously bleeding. And the next point, and maximum 50% of this disease is transmitted from the carrier mother to their son. Okay, and the last point, this disease is one of the example for crisscross pattern of inheritance. Hope you all understood all the points about this disease. So it's a 3-mark question. So now we completed one 3-mark question. Then second disease is a color blindness. Second example for this ceiling within is color blindness. Okay, so here color blindness means one of the disease affecting the human being. Normally, retina is one of the important part of our eye. Okay, retina is one of the important part of our eye. And this retinal region consists of two types of cells, rod cells and cone cells. Here, cone cells only are responsible for the vision of color. So now we are seeing different types of color. Okay, so this color vision is only because of the presence of cone cells. So our retinal region consists of more amount of cone cells. So because of this presence of these cone cells only we can see different types of color, or we can identify different types of colors. Understand a few? And some genes are controlling the production of these cone cells. Okay, so some dominant genes are controlling the production of these cone cells. If these genes are involved in mutation, they will be in recessive condition. So the recessive genes unable to produce more amount of cone cells. So the person is unable to identify red, blue, green colors. Okay, red and blue, green colors. Understand a few? Understand? So hope you understood this part. So once I repeat, here this cone cell is the cells present in the retinal region of the human eye, and these cells only are responsible for the vision of color. Okay, to see the color, the cone cells are helping to the human eye. Understand? And the genes controlling the production of these cone cells. And if these genes are involved in mutation, they are unable to produce the cone cells. So, the affected person cannot or unable to identify red and green color, red, blue and green color. Understand a few? Understand? Then, this disease is transferred from one generation to next generation by two type of marriage. Okay, listen next point very carefully. Hereafter, we are going to study in detail this color blindness. Okay, so this color blindness disease is transmitted from the father or from the mother to their son or to their daughter by two types of marriage. Okay, so listen very carefully. So before going to study about this disease, you should remember one important point. What type of persons can produce color blindness son? What gamete are responsible for the color blindness daughter? Or what gamete are responsible for the carrier daughter? Okay, so you should understand what types of gametes are responsible for some types of persons. See, look here. See, X, Y. Okay, so these X and Y, these two are the chromosomes present in the male. So, when the X chromosomes are in plus condition, Y chromosomes are in minus condition, that person is a normal man. Is a normal man. Then X plus and X plus. Okay, so it's a normal female because X and X, both X, females have two X chromosomes. Okay, so when these two X are in plus condition, that female is a normal female. When one X is affected, here affected means here I mentioned the letter C. Okay, C meaning color blindness. Here the letter C indicates color blindness. Okay, so here two sex chromosomes are there. One is the one X chromosome and next sex chromosome also X chromosome. But one X chromosome is affected, color blindness, and the next X chromosome is normal condition. So this female is a carrier female. This female is a carrier female. Two X chromosomes are there, but one X chromosome is mutant. That means changed into color blind. Then next, one X chromosome is color blindness in condition and the Y chromosome is in normal condition. So X and Y both are the X chromosomes are male. So here the X chromosome is damaged. So that person is a colorblind male. That person is a colorblind male. Then next, two X chromosomes are there. It's a female sex chromosome, but both chromosomes are affected. So that female is a colorblind female. That female is a colorblind female. So you should understand this one. Then only you can clearly understand this colorblindness disease, transmission of this disease. Okay, so let's say repeat this one. X plus Y minus is the normal man. Then X plus is a normal female gamete. Then XC and X plus is the carrier female's gamete. Then XC Y minus is the colorblind male gamete. Then XC and XC is the gamete of colorblind female. Hope you understood this one. Then so now we are going to study about how this disease is transmitted. See, this disease is transmitted in two ways. Okay, two types of marriage. So the first marriage is when a normal female marry a colorblind man. This disease is transmitted to the next generation, to the son or to the daughter. So this is the first marriage, first type of marriage. And the second marriage is when a colorblind female or woman marry a normal man, this disease is transmitted. This disease is transmitted. So when a normal female marry a colorblind male, this disease is transmitted. Or when a colorblind female marry a normal male, this disease is transmitted. So hope you are understood. So now we are going to study about how this disease is transmitted through the first type of marriage. So the first type is between the normal female and colorblind male. Okay, colorblind male. So listen very carefully. So the normal female gamete is X plus is the normal female gamete, and the colorblind gamete is XC and Y minus. XC and Y minus is the colorblind male gamete. Okay, so first suppose if these two gametes are joined, what we will get? X plus and XC. X plus and XC. Okay, then if these two gametes are joined, what we will get? X plus and Y minus. X plus and Y minus. Okay, understood? So X plus XC is the gamete of X plus and XC is the gamete of carrier female. Carrier female. Then X plus Y minus is the gamete of normal male. Normal male. Hope you understood. So when a normal female marry a colorblind male, in the first generation, F1 generation, there is a possibility for getting one carrier female and a normal male. Understand? Then now we are going to study about what happening in the next generation, F2 generation. See, when this carrier female marry a normal female, normal man. Okay, when this carrier female marry a normal man, what will happen? See this end. So first you are going to write the carrier female's gamete. X plus XC. See the gametes of carrier female, and X plus Y minus the gamete of normal man. Okay, normal male. So X plus XC is the first gamete and second gamete of the carrier female, and X plus Y minus is the gametes of normal male. Normal male. See, listen very carefully. Suppose these two gametes fuse. What we will get? X plus and X plus. Okay. Then suppose this gamete fuse with this gamete. What will we will get? X plus Y minus. Okay. Then suppose this gamete fuse with this gamete. What we will get? XC X plus. Suppose this gamete fuse with this last gamete, what we will get? XC Y minus. XC Y minus. Now listen very carefully, dear students. X plus X plus. So it's the gamete of normal female. Normal female. Then X plus Y minus. It's a gamete of normal male. Normal male. Okay. Then XC X plus. XC X plus. It's a gamete of carrier female. Gamete of carrier female. And last XC Y minus. XC Y minus. Color blind male. Okay. Color blind male. So in the second generation, year 2 generation, what we are getting? One normal female, one normal male, one carrier female, one color blind male. Okay, so we are getting the young ones or the next generation people in this ratio. Understand a few? Understand? So hope you understood this one. So this is the first way of marriage. Okay, first type of marriage for transferring the disease. What disease? Color blindness. Okay, then now we are going to study about second marriage. Okay, so second types of marriage. So the second type of marriage is marriage between a colorblind female and a normal male. So the colorblind, the gamete of the colorblind female is XC, XC. Okay, then the gamete of normal male is X plus Y minus. X plus Y minus. Okay, so listen, the first generation, what is happening? This gamete is fused with this gamete. So XC X plus. Then this, suppose if this gamete is fused with this gamete, XC Y minus. XC Y minus. Okay, so XC is the gamete of, it's the gamete of colorblind female. Okay, colorblind female. And X plus Y minus. X plus Y minus is the gamete of normal male. Understand it? So the first generation we are getting, so A1 XC X plus. XC X plus meaning carrier female. XC X plus. Okay, XC X plus is the carrier female. Then XC Y minus. XC Y minus is the colorblind male. Colorblind male. Understand? So in the second type of marriage, the F1 generation, we will get one carrier female and a colorblind male. Colorblind male. Okay, then let's see what happening in the second generation, F2 generation. So marriage between the second generation, marriage takes place between the carrier female and a colorblind male. Okay, carrier female, XC X plus, colorblind male, XC Y minus. Okay, gamete, XC X plus. This is the first gamete, this is the second gamete. Here X is first gamete, Y minus second gamete. Okay. So in the second generation, marriage takes place between the carrier female and colorblind male. Okay, carrier female and colorblind male. So what happens? See, this gamete is fused. Suppose this first gamete is fused with this gamete, we will get XC, XC. Okay. Then suppose this gamete fused with this gamete, what happening? XC, Y minus. Then suppose this gamete fused with this gamete, X plus XC. Suppose this gamete is fused with this gamete, what happening? X plus and Y minus. Okay? Okay, see. XC, XC. It's a gamete of colorblind female. Colorblind female. Gamete of colorblind female. Okay? Then XC Y minus. XC Y minus. Gamete of colorblind male. Colorblind male. Okay, colorblind male. Then next X plus XC. X plus XC is a carrier female. Carrier female. Then X plus Y minus. X plus Y minus. Normal male. Normal male. So in the second generation, we will get one colorblind female, one colorblind male, one carrier female, and a normal male. And a normal male. Understand, my student? So this disease, colorblindness disease, is transferred from one generation to next generation, from the father to son, or father to daughter, from the mother to son or mother to daughter. Okay, it's transferred from one generation to next generation through two types of marriage. The first type of marriage is a marriage between the normal female with a colorblind male, and the second type of marriage is colorblind marriage between the colorblind female with normal male. Understand of you? Understand, my students? So I hope you all understood this colorblindness disease. This colorblindness disease. Okay. Okay, dear students. Now the example for Y-linked inheritance. So now we completed the inheritance of X-linked genes. Now the example for Y-linked genes. Example for Y-linked genes. So two examples we have studied for the example for X-linked genes. Now we are going to study about disorders for the Y-linked genes. Okay, Y-linked genes. Hypertrichosis. Okay, hypertrichosis is the one and only example for you explain your book. Here, hypertrichosis meaning this disease transfer from father to son. Okay, only from male to male. It will not transfer from female to female because it is a Y-linked disease. Okay, Y is the sex chromosome, only male. So this disease is transferred from male to male, from the father to son. Understand? Your hypertrichosis meaning is the disease. Okay, if anyone is affected with this disease, more amount of hair is growing on the pinna region. Okay, so pinna means this region is called pinna region. So in some males, pinna region consists of more amount of hairs. This condition is called hypertrichosis. It's called hypertrichosis. So this disease is transferred only from the father to son because the father only having the X chromosome, sex chromosome, X chromosome. Okay, so it's an example for the Y chromosome. Okay, so it's an example for the Y-linked disorder. Understand, my dear students? Understand? Okay, dear students, now we are going to study about another one topic. The next topic is karyotyping. Karyotyping. Here, karyotyping is the technique used to separate the complete set of chromosomes from a cell. You all know 46 chromosomes are there in our cells. All these 46 chromosomes are separated from the cells and we can take photograph of these chromosomes. Okay, so this technique is called karyotyping. This technique is called karyotyping. Then, preparation of this karyotyping. See, two scientists, Tijo and Lieber. Okay, Tijo and Lieber. Understand? So in the year of 1960, these two people really first explained this karyotyping technique with the help of the leukocytes. Leukocytes meaning is the blood cells, okay, or WBC present in the human blood. So to produce this karyotyping technique, to do this karyotyping technique, first they used one chemical. Name of the chemical is called colchicine. Okay, so they add this colchicine chemical in the leukocyte cells. Immediately, the cell division is stopped in the metaphase stage. You all know the prophase, anaphase, telophase, metaphase, like that. Many stages are there in the cell division. Okay, so when this chemical is added in the leukocyte, this chemical stop or arrest the cell division in the metaphase stage. Okay, so now all the cells are in metaphase stage condition. There is no further division. With the help of the microscope, electron microscope, the photograph is taken now. Okay, now photograph is taken with the help of the microscope. Okay, then all the chromosomes are cut from the photograph and arranged in a order. This is a karyotyping technique. This is a procedure for the karyotyping. So to do this karyotyping technique, first colchicine chemical is added in the cells. This colchicine stops the cell division in the metaphase stage. After that, the help of the microscope, photograph is taken. So from the photograph, we can easily identify the chromosome. Understand? Then the application of this karyotyping, that means the uses of this karyotyping. There are a lot of uses are there for this karyotyping technique. The first use is, this technique is used to identify the sex. When the fetus is developing inside the uterus of the mother, we can identify the sex of the fetus, whether it is a male fetus or female fetus, with the help of this karyotyping technique. First use. And the second use is, this technique is used to identify chromosomal aberrations. Chromosomal aberration meaning problem in the chromosomes. Example, in the lower classes you have studied, I think, deletion, duplication, addition, translocation. Okay, so these are the example for the chromosomal aberration. So these type of chromosomal aberration we can easily identify with the help of the karyotyping technique. Understand, my students? Then the third use is, this technique is used to identify the chromosomal abnormalities like aneuploidy. Here aneuploidy meaning in some cells, in sometimes if any failures occur in the cell division, number of chromosomes may be increased instead of 46, or number of chromosomes may be decreased instead of 46. Okay, so this condition is called aneuploidy. So this type of chromosomal abnormalities also we can identify with the help of the karyotyping technique. Okay, and this karyotyping technique is used in the evolution field, used to understand the evolutionary relationship between the species. And the last use is, this karyotyping technique is used to identify the genetic diseases. So, dear students, now we have completed 5 applications of this karyotyping technique. First one is gender identification, used to identify the sex of the fetus. Second one, it is used to identify the chromosomal aberrations. Chromosomal aberration, example, addition, deletion, translocation, duplication. These are the example for the chromosomal aberration. And the third application is it is used to identify the chromosomal abnormalities. Okay, abnormalities meaning here what is an aneuploidy. Okay, and the fourth one is it is used to understand the evolutionary relationship between the species. Evolutionary relationship between the species. And the fifth one is it is used to understand the genetic disease. It is used to identify the genetic disease. Understand, my dear students? So hope you all understood the karyotyping definition for karyotyping and the method of karyotyping technique and the applications of karyotyping. Okay, so everything are important for 2 mark and 3 mark question. Okay, my dear students, today's class we have completed. The first topic is what topic we studied? The sex-linked inheritance. Example for X-linked inheritance, example for Y-linked inheritance. For example for X-linked inheritance, 2 examples we studied, hemophilia and concubinage. And the example for Y-linked inheritance, we studied about 1 example, hypertrichosis. Then finally we studied about karyotyping technique. Okay, so today's topic is very important for 5 mark, 3 mark, 2 mark and 1 mark question. For example, which chemical is used to stop the cell division during the metaphase stage, coalescing. Okay, like that many 1 mark questions also there. So today itself try to complete these questions. Thank you. We will meet in the next class.